Hey everyone, welcome back. So far, we have learned how to handle individual sequences. But in biology, we rarely look at sequence in isolation. We usually want to compare it to something else, like to find a mutation, identify a species, or see how a gene has evolved over millions of years. So this process is called a sequence alignment. So today we are going to use BioPython to align two sequences and learn the crucial difference between global and local alignment. So let's get into it. So what is a sequence alignment? Imagine if you have two strings of DNA that are similar but not identical. So to compare them, we line them up to see where they match and where they differ and where one might have an extra base like an insertion or a missing base like a deletion. So we use a scoring system like +1 for a match and -1 for a mismatch and a penalty for creating a gap. So the higher the score, the more similar the sequences are. So global versus local and when to use which one. So there are two main ways to do this. First is global alignment. This tries to align the sequences from the very beginning to the very end. It's best when the two sequences are roughly the same length and you expect them to be similar across the entire length. So global alignment and local alignment. This looks best for the best subset or patch of similarity. It is perfect when you have a small gene and you want to see if it exists somewhere inside a much larger chromosome. So BioPython makes both of these very easy to compute. So let's start with global alignment. So we will import the pairwise aligner from BioPython. This is the modern high performance way to do alignments. So for that we will do from bio import align. Okay, now we have defined two similar sequences over here. Then we will create an aligner object. Aligner is equal to align dot pairwise aligner. Okay, now we will perform global alignment. Alignments is equal to uh, aligner dot align seq1 seq2. Okay, now after that we will look at the alignment results. So best alignment is equal to alignments 0. You should know that Python index starts with 0, so we always use 0 as the first element. Print best alignment, then print global alignment score, mm, best alignment dot score, right? Yeah, okay. So let's see and try to run it and see what happens. Okay, so do you see how BioPython adds those dashes? Those are the gaps. It is shifting the sequences to find the maximum number of matches, and the score tells us how mathematically good this alignment is. Okay, so the global alignment score is 3.0. Okay, now what if we have one very long sequence and one very short one? So for that we will have to switch the aligner to local. So this section will be for local alignment. So let's define a long sequence, this, and short sequence. We can make a similar short sequence. GCTA, GCTA, G. Right. Now we have to set the mode to local. Right. Now, similarly as the last time, local alignments is equal to aligner dot align long sequence, short sequence, and best local is equal to local alignments 0. Then we print it, print alignment score best local dot score. Let's run this, and this is what we see. So, in local mode, the aligner ignores the parts of the long sequences that don't match and just zooms in on the perfect subsegment. So, this is exactly how tools like BLAST work when you search for a gene in a database. So, now let's try customizing the scoring. So, bioinformatics is all about the details. So, sometimes a gap is more expensive than a mismatch. So, we can customize our aligner to reflect a real biological reality. So, what we will do, aligner dot match score to aligner dot mismatch score -1, aligner dot open gap score minus 0.5. Now we will rerun the alignment with these values. Custom alignments is equal to aligner dot align sequence 1, sequence 2. Then we will print custom alignments 0. Right, let's try and run this. And here it is. By adjusting these scores, you can fine tune how the algorithm handles evolutionary changes. In a real lab, these parameters are chosen based on the type of organism you are studying. So, alignment is basically the bread and butter of bioinformatics. So, today we learned the theory of global and local alignment and used BioPython's pairwise aligner to visualize and score these comparisons. So, this is the foundation for everything that comes next. So, in our next video, we are going to take a break from sequences and look at data visualization. We will learn how to plot things like GC content and other things. And if you found this tutorial helpful, please give the video a like and hit the subscribe button. You can find the notebook with these alignment examples on my GitHub soon, and I'll see you in the next video.